Hello everyone, welcome to Echoes by Jigyansa. Today we have the privilege of hosting Dr. Nagaraj Guru Prasad, an esteemed professor at IISER Mohali and a leading evolutionary biologist. Dr. Prasad has done his PhD from JNCSR Bangalore and his postdoctoral research at Queen's University, Canada. His academic journey reflects both depth and global impact. Dr. Prasad's research explores fascinating areas such as sexual conflict, sexual selection, life history evolution, and the evolutionary ecology of immunity, primarily using Drosophila as a model system. His work helps uncover how evolutionary forces shape aging, reproduction, and immune responses. Beyond research, he plays a significant role in shaping the scientific community, serving on the national expert committees for major research grants and fellowships, and as a chairperson of the National Committee for the International Union of Biological Sciences. We are excited to dive into these thought-provoking ideas with him today. So let's get started.
Sir, the first question is, could you explain about your research in a couple of minutes?
Sure. My first love in terms of research has always been life history evolution. I'm very fascinated about what determines how an organism distributes its reproductive output throughout its lifespan, what decides its lifespan, what affects the entire life history. Those kinds of questions I have always been interested in. And my PhD was looking at life history evolution. What I was trying to do then was to look at pre-adult time and how does this pre-adult development time affect adult characters. Right? So I did selection for pre-adult development time. We create probably what is today the world's fastest developing flies. They are like supercars, and they have taught us many things about life history evolution. So yes, life history evolution is something that I really love to do. In my postdoc and much later on, I have been fascinated with intersexual conflict, both intra-locus sexual conflict and inter-locus sexual conflict. Intra-locus sexual conflict where you have expression of a certain allele in one sex, increasing the fitness of that sex but decreasing the fitness of the other sex when expressed in the other sex. So, which keeps the genome in sort of a tug of war between the males and the females. And that has very interesting consequences in terms of how gender is expressed in males and females. And how additive variation for fitness itself is maintained in populations. Interlocus sexual conflict, where males and females evolve with respect to each other, almost very similar to how a host and a pathogen would co-evolve. So you will see antagonistic co-evolution between males and females. Evolution in females drives evolution in males, and evolution in males drives evolution in females, but this coevolution is antagonistic in nature with respect to their fitness. And this is again very interesting for me because interlocus sexual conflict rapidly drives speciation. So therefore I am very interested in it. The other thing that we have done over the years is to try and look at how immune responses evolve in populations. Again, immunity is a very important component of any organism's fitness. And immunity has been studied in great detail in terms of the genetics and the molecular biology of it, especially using single gene mutants. Now, single gene mutants are very great if you want to work out specific pathways. But if you are interested in immunity as a life history character, then single gene mutation studies are not very useful. So what we have done is we have done experimental evolution studies. We have evolved populations to be more immune to certain kinds of infections, and then we have asked questions about what are the underlying mechanisms, what are the genetic changes, what are the life history trade-offs, and so on. So in some sense, you can think of life history evolution as a single theme that underlies all my research, and overlaid on that are things about interlocus conflict, immunity, etc. all using Drosophila melanogaster as the model system, and experimental evolution, which is painful but probably one of the most powerful techniques ever as a method of research.
The second question, from a biological perspective, what specific process, systems, or mechanism are you trying to understand? Are you looking at their behavior, evolution, genetic regulation, cellular processes, or something deeper?
Question is very broad. I want to understand evolution as a process. Right? Let me try to oversimplify it. Most of the times when I walk into the first class and talk about evolution, all the students are thinking of dinosaurs. Okay, I'm not interested in dinosaurs per se. I'm interested in the process that gave rise to the dinosaurs, or to humans, or to chimpanzees, or anything. I want to study evolution as a process, and I want to study evolution as a process dynamically in real time. Right? How do I do that? I can't do it in the wild. You can observe evolution happening in the wild, but it is difficult, and it is by chance you end up observing evolution happening in the field. The famous case of the beaks of the finches, right? People were not there to study the evolution of the beaks of the finches. They were doing some other study, they were measuring many other characters, and as a byproduct, they saw how the beaks evolved. And I don't mean Darwin study, these are the more recent studies, right? And it is difficult to isolate the sources of selection that cause evolution to happen if you are studying in the wild. Plus, maybe you are lucky, maybe you will observe evolution happening in the wild once. But how do I separate out my results from chance? I want to see evolution happening repeatedly under identical conditions if I want to isolate the process and be confident of this is the process that has resulted in this, right? So I am now left with doing evolution in the lab. So therefore, to address questions about evolution as a process, I do experimental evolution. I cause evolution to happen in the lab. I have replicate populations which are maintained under identical conditions, either be controls or be it selected populations, so that I can see evolution happening repeatedly in independent populations under control conditions. Remember that since evolution is happening in my lab, I can decide the time scale on which I am going to study it. I can study it every generation, every 5 generations, 10 generations. And the questions that you asked, I can decide the biological level of organization where I am going to study it. I can look at the behavior of the flies, I can look at the biochemistry, I can look at the tissue, I can look at gene expression, I can look at how SNPs are changing. That is the power of experimental evolution.
That's fascinating. What originally drew you towards this particular research area? Was there a moment or experience that sparked your interest in this field?
Okay, so I don't have a very inspiring story to tell you that, okay, right from my childhood I was fascinated by evolution. That is not true, okay. I was not even sure that I would become a scientist. I liked figuring things out, and I credit my people at home to have instilled that in me. I remember my grandfather, he was a very well read man. He was a high school teacher and a headmaster, and if I went to him saying that, oh, I have this question, what is the answer? He would never give me the answer. He would say, you know, we have this book, set of books, and you look at this book and this book and this book, and you look at this chapter, that chapter, you read it, and why don't you tell me what you think the answer is? This was his way of giving me an answer, and that taught me how to look for answers. And at some point of time I started enjoying figuring answers to questions. That was true, but it was not specific to science. I had questions of all kinds of things, and he would guide me along all kinds of things, right? Be it art or history or music or science. So figuring things out was there. Probably I got more and more into science during my college. I should give credit to my undergraduate college where we were allowed free access to our lab, especially our zoology lab. We could do whatever we wanted. We did all kinds of fun things like grow earthworms and look at their chromosome. Why did we do it? We have no idea. Caught flies from all around. Why flies? Not that I knew I would work with flies, because they were the easiest to catch. Or brought some fertilized toads' eggs and put them under a microscope and sat there till late in the night. Our professors, instead of going home, sat with us and allowed us to see it, and they also saw. So a lot of credit also goes to my college, where they encouraged figuring things out within science. When I joined my PhD also, I was not sure that I am going to become a scientist. To be honest, I joined PhD because I was told that if you wanted to become a teacher, to get a permanent job as a teacher, you should have a PhD degree. Right? So my focus was very, very small and very, very local. Then I applied for PhDs. Then I got into some places, and I was called to come and meet this person in JNCSR who I did not know. I went, I met him, and then I started talking to him. And over the next 1 or 2 hours of conversation, my mind was made up that I was going to join him, and I was going to do PhD with him. And in those 2 hours, we did not talk science only. We spoke about everything: music and movies and Kannada literature and Urdu poetry and some science also, all kinds of stuff. So I joined this person for doing my research because I liked the person without knowing too much about what was the research that he was doing. Okay, and over the first few months of my research, I actually fell in love with the research, and that's when I realized, no, this is what I want to spend my life doing: being a scientist, doing experiments, figuring things out, and so on. So in that sense, there is no one single moment, but there are important goalposts or signposts which I can in hindsight look at and say, okay, maybe this influenced me. Definitely meeting with my future PhD supervisor was one such moment. Being in the lab and figuring out what research was another such moment. There was some interest in genetics, that I was sure, because in my master's I had to take a special subject in my second year, and I wanted to take genetics. And in Bangalore University, where I studied, they used to teach cytogenetics, more classical genetics. So I had taken that, and there was some inclination to evolution as a subject, because both the years we were supposed to give a seminar, and we were free to choose whatever subject we wanted to. And now in hindsight, I realized that first year I picked origin of chordates as my seminar topic, and the second year I picked genetics and evolution as my seminar topic. But this is all post facto reconstruction, right? So if you had asked me sitting in my MSc class whether you will spend your life looking at evolution as a research topic, probably I would have said, I don't know, right? So, yeah.
Biology is moving incredibly fast with technologies like genome editing, high throughout sequences, and single cell analysis. What recent advancement have most excited or impacted your work?
Um, Let me answer it in two parts. Certainly, advances in technology allow you to do things and look at things in certain amount of detail that you were not able to do so earlier. That is true. However, it does not necessarily lead to greater advancements in the fields. Many a times, very simple experiments, very simple theoretical formulations, without the aid of heavy investments or super end high end machines, advance the field greatly. Right? In my field, evolutionary biology, I think greatest intellectual leaps in the field have happened without the aid of very high end technology. That does not mean that I am discounting the exciting new things that we have seen because of technology. For example, we have all figured out over the years how humans came to be what we are and how we got distributed around the world, the whole out of Africa and all that. We knew this story. We knew that this was more than a story. And we had figured this out by looking at archaeology, paleontology, linguistics, etc. But now, with the newer techniques of ancient DNA, you can add additional evidence to it saying that look, this what we had predicted, there is greater evidence for that at a genetic level. Plus, we also now know something that we did not know earlier. For example, that we have some Neanderthal DNA, which means that our ancestors interbred with Neanderthals or Denisovans. That we would not have figured out using linguistics, for example, right? So there is always some details which get added. They are beautiful, they are important, they open up newer areas, but big intellectual leaps, I don't think always require technological advancement. Right? So same it's with our field. The newer technology has brought in some newer perspective, newer details. Intellectually speaking, advances have always happened because of people bringing ideas together, people doing simple experiments.
What originally AI and machine learning are reshaping biology, especially in areas like protein structure, prediction, image analysis, and genomics. In your field specifically, where do you see AI contributing meaningfully? And what skills students should build to read?
Um, I do not know because see, protein folding and other things depend on certain basic laws of physics and chemistry. When you have an entire protein, figuring it out and putting it all together and figuring out how the protein folds is difficult so far, but AI and other methods have made it easier. If you think about evolution, predicting long term evolution, unlike predicting protein folding, is much more complex. Why? Because if you think about it, you have a set of variances and covariances. Then you have a set of selection pressures. When these selection pressures act on variances and covariances, you get some output which you think of as an evolutionary response. If it was only this and simple, then you didn't even need AI. Simple pen and pencil or pencil paper calculations would have told you what your evolutionary response would be in the long term. The complexity in evolution happens because with each iteration of generations, right, you are changing even this matrix of variances and covariances. So your predictive ability, the thing that helps you predict itself, is evolving. Evolution requires a matrix of variance and covariance, but evolution also changes this matrix of variance and covariance, and therefore long-term prediction becomes difficult. I do not personally think of any way in which just by using more recent advancements in computation or AI, you can predict the way this variance covariance matrix is going to change. Therefore, I do not believe that the same magnitude of advancement that you get in, let's say, protein folding, you will get in predicting long term evolution using AI is possible.
What is your view on current India's position in biological research, and what strengths do we have, and what major gaps must we address to accelerate world-class innovation?
Okay. Um, there are two different scales of biology. You can think of biology at a organismal and above organismal scale, and biology at a cellular and molecular scale. If you think of biology at the cellular and molecular scale, it is addressing certain kinds of questions which are more proximate mechanism based questions. These are important, these are interesting. If you look at biology at the organismal and above organismal scale, you are addressing questions of the ultimate nature. Why, in the sense of evolutionary why questions. Right? For doing cellular and molecular kind of work, by the nature of the work, you require large sums to be invested in terms of money for your chemicals, for your machines, and so on. Right? And this is the place where India is still not self sufficient. Um, when you are working in those kinds of areas, you are competing with investment abilities of people outside versus investment abilities of me, in addition to your own ability to think, right? And many a times our thinking ability is as good, but our investment abilities don't match up. So quite often we can't become the world leaders. There are of course small pockets where we are world leaders even in this kind of biology, but in general our investment ability is low. But if you look at other kinds of biology, the organismal biology, if you think of evolutionary biology or behavior or ecology, there a lot of the research is not resource intensive. It is my idea versus your idea. It is my brain versus your brain. It is not my money versus your money. This is the area where you can actually match with anybody else in the world on equal terms. And Indians do match others around the world in equal terms in such areas. In fields of evolutionary biology, in fields of behavior, Indian scientists are considered world leaders. Right? And we do set the agenda. We are part of debates about what constitutes evolutionary biology, what are the topics that are important, what is the view on various issues within evolutionary biology. Indian scientists are actively involved. You asked about long term plans and what should be done to improve. We have been investing more and more and more money. That is good. We should try to invest more. We should try to bring in our technological superiority or technological levels or ability to make chemicals, etc., to the same level as the other thing. But more importantly, we should learn to play to our strengths. Our strengths is the ability to think. That is second to nobody in the world. So therefore, instead of focusing on only certain kinds of biology in universities and syllabi across India, you should learn that there is a whole different kind of biology which is important and where we can succeed. Therefore, across India, it is important to re emphasize the teaching and learning of areas like evolution and behavior. That is easily done. That doesn't require monetary investment, right? It just needs you to re emphasize what we have been good at for a very long time and traditionally. So, what I think needs to be done is to take a conscious step in putting these kinds of biology on equal footing. That is what needs to be done. We have to learn to play to our strengths.
Hopefully, India will do this in future.
Yeah, I mean, see, for example, if you look across India, how many institutions of cellular and molecular biology exist? Lots, lots. How many institutes of evolutionary biology exist? Zero. There is no National Institute of Evolutionary Biology, but there is National Institutes of various kinds of subcellular biology. Why can't we establish one? Why can't we create one umbrella organization where the best minds of India are put together and increase the superiority that we already have? We could start by doing that.
For students and young researchers who want to pursue science seriously, what mindset, skills, and habit would you say matter the most today?
Um, if you want to pursue science, it is best done in the amateur spirit. Science should not be seen as another career. It should not be treated as another career. Right? You do science because you enjoy doing science. It is not something that you do because it pays your bills. Of course, it should pay your bill, but you cannot do it as just another career. You have to do it because you really love it. It is in that sense I say that there should be an amateur spirit. Of course, Professor Joshi puts it differently. He says that it should be done in the Sufi spirit. So in that spirit is what one needs to do science. If students get out of this idea of science is a career, but I will do science because I love doing science, that helps a lot.
That's very true. Okay. One final fun question. How was Bodhi sir during his student days? Is there any particular memory you would like to share?
See, Bodhi was, and you might be disappointed because I'm saying this, because he was a very sincere student. Very sincere student. There are no juicy stories about Bodhi. Not because I'm trying to hide it, because there really are no juicy stories about Bodhi. Um, Bodhi came to, okay, what is the best memory I have? There are many such ones, but I think here is one that is very important. The first time I met Bodhi, I was in a shared office space, and I came back from a class and I see this young boy sitting, waiting for me, and he said, "My name is Bodhisatta Nandi. I want to speak to you." I said, "Yeah, let us speak." Then he said, "I am from this college. I have done my master's in zoology, and I want to join you for a PhD." Then I told him that, "Look, oh, very nice. You are obviously bright," after having spoken to him for 10 minutes. "But I advise that you don't join me for a PhD, because doing PhD in evolutionary biology using Drosophila is very tough. So let's forget it. Okay. Don't join, do something else." And then he was sort of confused, and then he left. Then the next day he came back again. He said, "No, I want to join." And then I told him again the same thing. "No, no, no, this is too tough. Why join? Do something else." He again reluctantly left, and then he came back again. I said, "No, I want to do this. I am convinced. I want to do this." And then I said, "Okay, join, and we'll do something." And he joined, and that was probably one of the best decisions that I ever made. We had nothing. We had to start everything from the scratch. He helped me build my lab. If you look at it, we are about 9 years apart in age. That's all. And so it was more like a younger brother. He worked with me. He worked with me to establish the lab, and we had many, many funny instances of establishing the lab. We were trying to cut costs. One of the basic things that we need to grow flies are cages, and getting cages was very costly. So we said we will buy food boxes, and you need to tie a cloth sleeve so that the flies don't escape. And you can't do that for food boxes, like the boxes where you keep your food grains and coffee powder and all that. So we decided that we will go and buy stockings. And if you put stockings on the mouth and cut off one end of the stockings, then you have something that will fit tight, and then you can put the flies in and out. So we decided to go and buy stockings. So me, Bodhi and Imrose, 3 boys, right, one older one and 2 younger boys, walking into a clothes shop and asking for stockings. That too in a very traditional city like Kolkata. And then this shopkeeper is looking at us, ki, why do you need stockings? Oh, we want to experiment. He is more worried. And then how many do you need? 50 pairs. He is getting very worried. And then we realized what was happening. So then we had to sit down and explain to him and then get the stockings. So yeah, lots of such fun things we had to go through. And when I moved from Kolkata to Mohali, he moved again. And then we established a lab in a unused electrical substation. There were no roads, there was no electricity, there was no place for him to sleep. He would literally sleep in the lab, and our lab was run with a electrical generator, the ones that you use in marriages, right? That kind of it. He trained how to start and stop that electrical generator. Lab was in the middle of wilderness. I still have that photo, maybe I'll share with you. One day I was in Bangalore, he sent me a photo. And all I could see was 2 bright spots. And I asked him, what is this? And this is middle of the night, what is this? Then he tells me, sir, that is a snake. Where did you find the snake? It was wrapped around my leg. What was happening was our lab was in the middle of nowhere. In the night, he was doing night checks, and then he felt something cold on his legs. And then he saw what it was. It was a snake which had come in, and then of course he shooed it away. So he has had many such experiences. But he set up the scientific rigor in the lab. He started the culture of bringing in undergraduate students and training them, not treating them as just a pair of hands, but treating them as human beings. He was genuinely interested in their welfare. He helped them, they helped him. He set up this very nice mentoring culture in the lab, which continues to this day. And his standards of doing research was excellent. He was, and he still is, a brilliant scientist. So I was lucky to have him as my student.
Thank you, sir.
Thank you.
It was very nice talking to you.
Same here. Nice talking to you.